Okay, as usual, I'm just going to let this load. It's loading. So we're going to talk a little bit about enzymes in more detail. So again, according to your Mr. Anderson notes, that was basically just to kind of introduce you to the idea of enzymes and how they function and just kind of get a general idea. So remember that the substrate is the thing that the enzyme is acting upon. An enzyme, of course, is a type of protein. The substrate fits into the active site, and then we'll turn that substrate into products. In chemical reactions, substances are changed into different substances by the breaking and forming of chemical bonds. So, when we talk about breaking bonds, we're talking about hydrolysis. When we talk about forming bonds, we're talking about dehydration synthesis. So, those two processes are driven by enzyme reactions. So, let's just look at a general equation. This is the equation for cellular respiration. So, we're going to take some carbon dioxide and oxygen, those are the reactants. Reactants are the substances changed by the chemical reaction, just a teeny bit of chemistry vocab review. The arrow represents the direction of the reaction, and we say that that yields. And then the area number 3 are the products, the substances that are formed during chemical reaction. So we take glucose and oxygen, then enzymes facilitate this chemical reaction that releases energy and also CO2 and water. Bond energy is defined as the amount of energy required to break apart a mole of molecules into its component atoms. That's chemistry, which is fine. I appreciate it, but I'm not a fan. Anyways, we're basically just talking about how bonds are sources of energy. We store energy there due to the way that electrons interact. And so when we break a molecule off, we release energy. If we put a molecule in, we're storing energy. So there's two types of reactions. There's an exothermic reaction. And so AP wants you to kind of be able to read these components of an exothermic reaction graph. And so you can see that the reaction progresses from left to right. Spot 4 would represent the reactants in the reaction. 5 is the activation energy. I'll talk more about that in a second. 6 is the total energy released. And then 7 represents the products. You can see that the products are lower than they were at the beginning in the graph. That's because energy is released. And so in an exothermic reaction, the main thing to know is just bullet number 1. More energy is released than absorbed. It's usually released as heat or light. And the reactants have a higher bond energy than the products. But again, just knowing that energy is released in an exothermic will be sufficient enough. The other type of reaction is an endothermic. And again, it's a very similar graph, but there's a slight difference there at number 11. So 8, again, the reactants. 9 is the activation energy. 10 is the total energy. And 11 are the products. So activation energy is just the amount of energy you need to get the reaction started. It's always there. It builds up, builds up, builds up until a reaction happens. In an endothermic reaction, again, the first bullet's the main thing. More energy is absorbed than release. And the products have a higher bond energy than the reactants because we're actually storing energy. So I know you're thinking, big whoop, Miss Ellis, why should I care? Well, this is why you care. A catalyst lowers activation energy. Activation energy, the amount of energy required to start a chemical reaction. It's a catalyst, decreases activation energy. Catalysts decrease activation energy for a chemical reaction. They will increase the rate of the chemical reaction. They're not reactants nor products, so they don't change, nor are they used up. They're there to promote this reaction to happen much quicker. So when a catalyst is present, less activation energy is needed to start a chemical reaction. So let's put this back into biology context. So a catalyst will decrease activation energy and increase reaction rate. It's not used up during a reaction and it does not alter the equilibrium of the reaction. And you can see there from the graph, the blue shows when you're not using a catalyst, the red shows when you are. So it significantly decreases that activation energy. Enzymes like catalase or protease, or lipase, which break down fats, or amylase, which is present in your saliva, and that's the first thing that starts to break down sugar as soon as you eat it. Enzymes are catalysts for chemical reactions in living things. So from now on, we're going to replace catalysts with enzyme. They're almost all proteins, like 99.9%. I could Google one that's not a protein. I don't really remember off the top of my head, and you're welcome to, but trust me, they're mostly, like, let's just say for all intents and purposes, they're proteins. And they almost always end in ASE. So just like sugar always ends in OSE, enzymes always end in ASE, and they're typically named after the substrate that they're acting on. One tiny exception, pepsin. Pepsin does not end in ASE, but it is nonetheless an enzyme. So homeostasis in enzymes. Homeostasis is just the constant set of internal conditions in an organism that need to be maintained. Temperature, pH, that kind of thing. And so large changes in temperature or pH can cause enzymes to break down or to denature because the hydrogen bonds will break. So for instance, let's say that you get a fever. A low-grade fever is okay. Low grade will be like 99, 100, that your body needs to do that in order to fight off infection. However, if that fever starts to get up into the 102, 104, 106, people often die from organ failure. Well, the organ failure is due to the fact that enzymes are beginning to denature and no longer functioning, causing an organism to die. So, enzymes function best at specific temperatures and pHs. Now, those can vary, but they have optimal pH and temperatures that they function in. And so, stable conditions are necessary for an organism's enzymes to function properly. Some of my students get confused and they think that enzymes maintain homeostasis. I mean, there's a lot of things that maintain homeostasis, feedback loops, which we're going to learn about later. But really, the idea is that enzymes function in stable conditions. Of course, you know that every protein structure determines its function. And so if you change the function, you're changing the structure. So altered enzyme structure can prevent an enzyme from functioning properly. That's actually something you're going to explore after this. So let's talk about substrates. Substrates are the reactants acted upon an enzyme. So all we're doing is just replacing a term you already know, reactions or reactants, what you start with in a reaction. We're just changing that to substrate for biology. They bind to an enzyme's active site, as shown in the diagram. Scientists like to use the analogy of a lock and key. Again, students sometimes think that we're saying that a certain substrate will unlock a certain enzyme. That's really not it at all. What we're doing is we're using the lock and key model to describe the specificity of enzymes and substrates. So only a particular substrate will bind to a particular enzyme, or flip it around and say enzymes only act on certain substrates, right? So sucrase will act on sucrose, the sugar, but it's not going to act on lactose. Okay, so we're talking about the specificity with this lock and key model. When bound, enzymes weaken the bonds within the substrate and allow reactions to occur. And these reactions would eventually take place, like eventually, but enzymes speed up the process by decreasing that activation energy. We need these reactions to happen fast, and enzymes take care of that. Okay, so I'm going to present you with some like thinking questions, processing questions, and I want you to just kind of put your notes away for a second, maybe find a scrap sheet of paper. Obviously, you're not going to turn this in. This is my in-person presentation. But I want you to think about these 4 questions, and then I will come up with a way for you to check your answers a little bit later. Okay, so first question, describe the relationship between enzymes and reaction rate. You want to write a complete sentence, kind of how are those things related? Describe the relationship between enzymes and homeostasis. Describe the relationship between enzymes and substrates. and describe the lock and key model as it relates to enzyme function. Now, the idea is I don't want you to like write directly off the notes this presentation, but if you can't respond, you can review the information, but you don't want to just write directly off that. All right, that does it for enzymes. Thank you. And I will let you know how we're going to check the answers to these prompts.